Okay, so now we're moving on to chapter 9, and just like chapter 27 and 28, we're just going to cover the surface of this chapter. We're not going into a lot of depth. So 29, we're going to talk about heredity. So just a couple of terms. When we're talking about genes, we're talking about segments of DNA, and these segments contain the blueprints for the synthesis of proteins. So the whole purpose of having DNA is to undergo transcription into RNA and then translate RNA into proteins. So the whole reason why we have DNA is to make proteins, and that is the central dogma of biology. How we control which genes we express or turn into proteins are actually going to be controlled by other genes. So we call this study of heredity genetics. And the basic principles of genetics were proposed by Gregor Mendel, who studied inherited characteristics that were either all or none, and he used pea plants for this. When we're talking about our human traits, though, they're much more complex than the original genetics principles that Mendel proposed in the 1800s. There is the Human Genome Project that has determined the human DNA sequence, which can now aid in genetic research and genetic screening. They're working on genome products for a variety of different organisms, including bacteria and viruses, just so that we can go back in and reference them. And that's a whole new field, which we actually call bioinformatics. So the vocabulary of genetics. So when we're talking about humans, we have a diploid number of chromosomes. So we have 46 total chromosomes, but these 46 chromosomes are in pairs. So we have 23 pairs of what we call homologous chromosomes. This is all of our cells except for our gametes, so all of our somatic or our body cells. When we're talking about the 23 pairs of homologous chromosomes, one set, so 23 chromosomes, come from the father, and 23 chromosomes come from the mother. And they're homologous chromosomes because they look very similar, and they carry genes for the same traits. But the traits are not equally expressed depending on whether it came from the mother or the father. So of these 23 pairs, one pair, so 2 chromosomes, are sex chromosomes that are going to determine the sex of us. So for XX, if we have 2 X chromosomes, you're going to be a female. If you have an X and a Y, you're going to be a male. And then the other pairs of homologous chromosomes that we have are what we refer to as autosomes. And this is basically going to be responsible for all of the other traits that we have within an organism. We do have something called a karyotype, and this is basically displaying all 23 pairs of homologous chromosomes that we have. And when we're talking about a genome, we're talking about our actual DNA or genetic makeup. 2 sets of genetic instructions. Again, we have one coming from the mother and one coming from the father. Here's an illustration. You don't need to know a lot of details on this slide, but I just wanted to show you this is a karyotype right here where we have organized our 46 chromosomes into their homologous pair. So here you can see all of the chromosomes into 23 pairs. Each chromosome within a pair means that one came from the mother and one came from the father. So we have gene pairs, again, within homologous chromosomes, and these gene pairs we refer to as alleles. And alleles are just different versions of the same gene, and they can code for either the same or an alternate form of that trait. So we can either say that our alleles are homozygous, meaning they are the same for a single trait. The DNA sequence would be identical on both of the homologous chromosomes. Or we can say the alleles are heterozygous, meaning they are different for a single trait, and the DNA sequence on the paternal and the maternal chromosome are different. So if we have 2 different chromosomes, sometimes we can have what's referred to as dominance, where one allele can go in and mask or suppress the expression of the recessive partner. So we typically denote dominant alleles with a capital letter and recessive alleles with the same letter, but we use the lowercase version of it. So for example, if we're talking about, let's say, eye color, and blue is, let's say brown, is dominant to green. So what we're going to do is we're going to choose the letter of the dominant allele. So if we have brown eyes and we have green eyes and brown is dominant, we would pick a capital B to represent our dominant brown allele, and we would choose a lowercase b to represent the green allele for eye color. Because again, we're using one letter and the only of the dominant allele, and then the capital version represents the dominant allele, while the recessive allele is going to be represented by a lowercase letter. A dominant allele does not mean that the allele has a higher frequency in our population, because sometimes we can have a dominant allele that is actually lethal and would not be found in a high frequency of the population. So again, we're just talking about in regards to dominance, if one allele can suppress the expression of another allele. Again, just some vocabulary for you. When we refer to a genotype, that is the genetic makeup of a person. So for example, when we did a capital B to represent a brown allele and a lowercase b to represent a green allele, that is the genotype. The phenotype is the physical expression of a genotype. So we would say the capital B represents brown eyes. So brown eyes is the phenotype, and a lowercase b represents green eyes. So green eyes is the phenotype. This is your genotype. So each person is genetically unique, and the reason why we are unique and we don't produce genetically identical offspring, with a couple of exceptions, is because we have events that occur during meiosis that contribute to genetically unique individuals. One of which is independent assortment, another one is crossover, and the third one is going to be random fertilization, and we're going to talk about these very briefly. So two important separations of traits occur in meiosis 1, more specifically metaphase 1 of meiosis 1. So when we're talking about segregation, we have two alleles of one particular trait that are going to be separated and distributed to two different daughter cells that we refer to as gametes. So for example, if we had alleles, capital J and lowercase j, one of the alleles would go to one cell, and the other allele will go to another daughter cell. So one of the daughter cells would get the capital or the dominant allele, and the other daughter cell would get the lowercase or recessive allele. So this is what we refer to as segregation when we're dividing up these alleles. There are errors that can occur during segregation where one daughter cell is missing the allele and the other daughter cell gets two copies of the allele. These can lead to various disorders, so things like cancer, infertility, or even Down syndrome. So we also have independent assortment, which was one of the factors that I said could contribute to increasing genetic variability. And we're working with the alleles of two different traits, like we've been talking about, that are on two different chromosomes. Well, these are going to be distributed independently of each other, meaning they are not connected. So if, for example, if we had a dominant allele and a recessive allele on one chromosome, and then we have a completely different trait, so a capital J and lowercase j on another chromosome, their assortment into daughter cells is independent of each other. It's not relying on another allele to go into a daughter cell in order for that particular allele to go into a daughter cell. So whether you inherit the dominant or recessive is of one trait, is going to be independent of whether you inherit the dominant or recessive allele of another trait. So that's independent assortment. So this is going to vastly increase our genetic variability. So just for an example, if we're talking about the number of gamete types that we have, since we're diploid, it would be 2 to a superscript of n, where n represents the number of homologous pairs. So technically, we could have 8.5 million combinations occurring just from our 23 pairs of homologous chromosomes. So this is just an overview. This is not us, because if we look at the image, we have 6 chromosomes where we're forming 3 different, what we refer to as tetrads. Okay, the tetrads of homologous chromosomes, they align independently of the other tetrads. So for example, we have different ways in which these can align up. So let's say the purple are paternal and the green are maternal. Well, they could line up just like this and assort independently, just down like this. Or they can line up like this, where we have one paternal, one maternal, and one paternal on this side. And on this side, we have one maternal, then a paternal, and then a maternal. So we can line up our homologous pairs of chromosomes on either side of what we call the metaphase plate. The next thing that it can increase genetic variability is crossing over. So we have genes that are on the same chromosome. They are linked and they can be passed to daughter cells as one unit. So during crossover, which occurs during prophase 1 of meiosis 1, homologous chromosomes can break, and even between linked genes, and we have what is referred to as a precise exchange of gene segments, and we have crossing over where they can go in and kind of exchange material. What we form after we form the crossover and exchange material are what we refer to as recombinant chromosomes. And recombinant just means that it has DNA or genetic information from 2 different sources. So now the chromosomes are a mixture of contributions from both parents, and again, we are increasing our genetic variability. So this is just an illustration showing you what's going on. We have a purple chromosome right here, and that's our paternal one, and then we have a maternal one here in green. Well, they form this chiasma where they kind of connect and link across, so we can see that in the image right here. Where they connect and where they touch, they can exchange pieces of genetic material. So we get something like this down here. So now the paternal chromosome has a segment or an allele from the maternal chromosome, while our maternal chromosome has a piece of DNA or genetic material from our paternal one. So at this point, we have formed our recombinant chromosomes, which again contain genetic information from 2 different sources. So when we go to make our gametes and we divide these homologous pairs into individual chromosomes, we're going to